In the interest of time, I think I'll start with the introduction. We have another great speaker lined up here. For the next session, Mirna Khatib received her PhD at the University of Florida and is now an assistant professor of biochemistry and biophysics, and actually won a recent career development award, so congratulations for that. And Mirna will tell us about flash mechanisms and alternative role of oxygen in the flash effect. Thank you very much. Join us here.
Thank you for the kind introduction, and thank you for the invitation. So today I will be discussing our work on oxygen measurements in radiation therapy, particularly in relation to the flash effect. The flash effect is composed of two parts. The first is that at ultra-high dose rates, we have less tissue damage in the normoxic region compared to conventional rates. The second is that the tumor control is equally maintained to the conventional rates. The cartoon here shows the flash effect in relation to oxygen. Oxygen is known to enhance radiation damage for both flash and conventional radiation. The difference in damage is higher for well-oxygenated tissue than for hypoxic tissue, and this is due to the oxygen effect. So oxygen plays a major role in radiation. The radiation results in the splitting of a water molecule into a hydrogen radical and a hydroxyl radical. The hydroxyl radical is a powerful and indiscriminate oxidant, which can rip off an electron from pretty much any organic molecule within its vicinity. For example, with DNA, it can generate a DNA-centered radical. This can be reduced back to the native form by the existing reducing species in the cell, or it can react with a molecule of oxygen, generating a DNA peroxyl radical. This is irreversible and is termed as fixation of damage. So obviously, the higher the concentration of oxygen, the more fixation that occurs. Quantitatively, this oxygen effect can be shown by the oxygen enhancement ratio curve. As you can see here, in well-oxygenated tissue, the tissue suffer or experience a threefold radiosensitivity compared to tissue in a more hypoxic environment. There have been several hypotheses trying to explain the flash effect. Amongst them, the most prominent and testable one was the oxygen depletion hypothesis, which essentially suggests that at ultra-high dose rates, there is fast and profound depletion of oxygen, therefore protection of normal tissue, unlike conventional rates where the fast resupply of the oxygen from the blood does not result in this deep depletion of oxygen. For tumors which are on the onset presumed to be hypoxic, the difference is less pronounced, and therefore tumor control is maintained. There have been several studies to date trying to measure oxygen measurements during radiation. Almost all rely on the phosphorescence quenching method. The phosphorescence quenching method essentially entails that oxygen quenches phosphorescence of molecular probes, such that the concentration of oxygen is measured by way of phosphorescence lifetime. So essentially, the concentration of oxygen is inversely proportional to the lifetime of the probe. So the higher the oxygenation, the shorter the phosphorescence decay that is observed. In our studies, we have used phosphorescent probes called oxyphores. These have been designed and developed in our lab. They are water-soluble, inert phosphorescent probes that report on absolute quantification of oxygen in biological environments, since their calibration parameters have been shown to be unaltered in such environments. Please note that these probes have been used previously in many biological studies trying to quantify oxygen. However, before these studies, they have not been used in regards to radiation. The first such study measuring oxygenation using phosphorescence quenching method was actually performed using solid-state sensors. This was done by Janssen et al. In 2021 in model solutions. The experiments done in vivo in the blood plasma using oxyphore probes were done in electron flash. As you can see here, that the results show that a single dose of flash radiation does not result in instant hypoxia. However, at the time, the time resolutions of these measurements were low to monitor oxygen dynamics during the application of the electron flash. So what remained as a question was, what if instant hypoxia occurs between the two consecutive measurements? However, we were able to measure oxygen dynamics using proton flash, which is much slower than electron flash. As you can see here, we were able to resolve oxygen dynamics during the application of proton radiation. And we were able to do such measurements at several kilohertz measurements. As you can see, flash rates result in fast stepwise oxygen depletion, where the oxygen concentration is proportional to the radiation dose as expected. We have performed these measurements in model solutions, in different model solutions, where we then determine the oxygen depletion G value. So that is essentially the concentration of oxygen depleted per gray of radiation. And what we obtained was two interesting results. The first is that regardless of the buffer used or the radiation delivered, that a single clinical dose of radiation does not result in instant hypoxia. The second key finding that we obtained was that regardless of the buffer used, on average, flash depletes less oxygen than conventional radiation. And we feel that this is key in understanding the flash effect, because if less oxygen is depleted, this could also mean less tissue damage and therefore radioprotection that is seen in the flash effect. So the fact that flash depletes less oxygen than conventional radiation is due to the recombination of primary radicals. Water radiolysis generates primary radicals, but these can recombine before they diffuse away from their particle tracks. Recombination scales quadratically with the radical concentration, and hence the dose rate. So as we increase the dose rate, we also increase the local concentration of primary radicals, which then causes an increase in the recombination, and therefore less oxygen is fixated. Another very interesting and unexpected result we obtained was the dependence of the oxygen depletion G values on the baseline oxygen. As you can see here, that we have a drop of the G value at already 30 micromolar concentration of oxygen. This is highly unusual because based on the abundant radiochemical literature, the rate at which oxygen reacts with organic radicals is diffusion controlled. So one should expect this drop at less than 1 micromolar and not 30 micromolar as we see. This result is very reproducible and was actually seen by others in the field. And we feel that it is significant in explaining the flash effect, as you will see in the future slides. Please note the resemblance of this curve to the OER curve that I showed you earlier. We have also performed time-resolved oxygen measurements in vivo using murine models. Two key things to note is that at conventional rates, We did not see any apparent oxygen depletion, and that is because there is a fast resupply of oxygen from the blood. Please note that this does not mean that there's no oxygen being depleted at conventional rates. It only means that at the rates that we're delivering the radiation, it's getting masked by the fast resupply of oxygen from the blood. The second key finding is the trace that you see here. This is the result from our radiation delivered at ultra-high dose rates for normal tissue. Again, a single clinical dose of radiation did not result in transient hypoxia in murine models. Another key finding that we observed was that there is, again, this dependence of how much oxygen is being depleted on the initial concentration of oxygen. This is exactly what was observed in our model solutions. So it can be very key in understanding the flash effect, because if oxygen depletion G values depend on the baseline oxygen concentrations both in model solutions and in vivo, well, then maybe we can explain the flash effect using oxygen dependence alone. If we assume tissue damage is proportional to how much oxygen is being depleted, so the oxygen depletion G values, which is the case, or it's indeed because the more oxygen, the more fixation that can occur, and therefore more tissue damage. And although we could not perform in vivo measurements at conventional rates to measure the G values, but if we assume that they are the same as those in the model solution, then the features of the flash effect really come directly from our oxygen depletion G values. And here shows this in a more picturesque way. So as you can see here, if we assume that the in vivo results match the in vitro results, and that flash depletes less oxygen in the normal region than conventional rates, and we correlate tissue damage to fixation, well, then that means less tissue damage is occurring. The difference at the more hypoxic region disappears between flash and conventional, and this is exactly what is observed in the flash effect. So less tissue damage is caused in the normoxic region because less oxygen is being depleted by flash, and therefore less tissue damage. But in the hypoxic region, where essentially the difference disappears, we see tumor control. So far, I had shown you the results in model solutions and in vivo using our oxyphores, where they were, did the measurements both in the blood and in the interstitial fluid. But since the damage occurs in the cell, and the cytosol is a different chemical environment than the model solutions and the blood plasma, it was important to make sure that does instant hypoxia occur in the cytosol, and we're missing something. So the probes that we have prepared report on quantification of oxygen. So they are designed in such a way that they do not permeate cellular membranes. So we can actually do the measurements in blood, in the tissue, but they do not go inside the cell. In order to overcome this, we use a well-known method by using electroporation, which is known to transfect cells with DNA. And we were able to incorporate our probes into the cytosol of the cell. As you can see here, the probe is uniformly distributed in the cytosol of the cell, where we can collect an emission, phosphorescence emission. But more importantly for these studies is that the calibration experiments confirmed that the probe's quenching parameters remained unaltered inside the cells, which is important because now we can actually quantify oxygen in the cytosol of the cell. These measurements were done in a cellular pellet to mimic the tissue. However, the one thing that we faced was because it's a compact layer of tissue, respiration is high. So by the time we walked with this cell pellet to the bunker, the respiration was all the way down to 0. In order to overcome this and slow respiration, we took advantage of a well-known chemical, which is potassium cyanide. Potassium cyanide is known to inhibit mitochondrial cytochrome oxidase. So this is what we observed. So as you can see here, please note, look at the inset graph. You can see clearly that in the absence of radiation, the cells are respiring. Upon application of radiation here at conventional rates, we get a steeper slope. And we can then determine the G values taking into the respiration factor. We have also performed these measurements at ultra-high dose rates, where as you can see here, inside the cytosol, we again see this fast stepwise oxygen depletion. The first key result here is that no transient hypoxia was again observed in the cytosol at ultra-high dose rates. The second key result that we obtained and was surprising initially for us was that the difference in flash at conventional rates disappears in well-oxygenated tissue compared to the model solutions that we had studied earlier. So since there's no difference in oxygen depletion between conventional rates and flash rates in cells, does this mean that all the oxygen-based hypotheses out there are nullified? Not really. So we do not have the full answer yet, but we have to keep in mind, like I said, the cytosol is a chemically different environment than the model solutions we worked in or the blood plasma. And most importantly is that the cytosol has detoxifying enzymes, which convert the superoxide and peroxides that are generated in the radiation back to oxygen. So although we don't have the full answer yet, I would like to show you a preliminary result that we obtained, that we went back to our model solutions and we added catalase, for example. And again, we don't see the flash effect anymore. We don't see the difference between flash and conventional rates. At this point, we have a lot more to study and to examine and to a consistent study with different enzymes to see what is occurring. But what I would like to do is thank the Penn team for doing these studies. I would also like to thank the funding agencies, especially the NIH, for their generous funding. Again, thank you for the invitation and thank you all for your attention.
Thank you very much. Are there any questions? Please come to the microphone.
Mirna, very nice presentation. I'm just going to argue against the oxygen depletion hypothesis that you bring up with the OER curve. Can you just bring it up? Probably it's easy if we just discuss on it.
You mean the first?
No, that one over there with the green and blue.
Here?
No, the next one. One more, I think it is. This one. So it makes total sense if you drop the oxygen from the normal tissue to the tumor, that you could argue that it's oxygen depletion, right? But my question would be, what if you double the oxygen? You go from 30 millimeters of mercury to 60 millimeters of mercury. In that model, there's no change. Yeah. But in Pierre's experimental data for the brain, doubling the oxygen reduced the flash effect by 1/2. So how would you go to the right, not to the left? Mm-hmm. What will happen if you go to the right of this?
So, a very good question. We have looked at higher oxygenation. Maybe I can pull it up. And there is still more to study, but we, let me just see if I can pull it up. No. Yes. So you're talking about if we go higher than...
so roughly 30 millimeters of mercury
so around in this area, basically.
It's lower, in fact. But
lower.
Yeah, so it goes from roughly 40 micromolar up to 60 micromolar. And that's where Pierre, for example, did his brain study 2 or 3 years ago in the brain, right?
Yeah.
And what he saw is if you go from 40 micromolar to 80 micromolar, that's roughly 4% to 8%.
Okay.
The flash effect was reduced by half. And in this case, it's not possible. You see, there's this fully saturated regime of oxygen. But we see that in vivo. So this is why I think the depletion hypothesis does not explain the high oxygen effect. It does explain potentially the low oxygen. I'm not arguing against that.
Yeah.
You're absolutely right. But if I start to increase oxygen, I start to reduce my flash effect. And this is the part that I don't understand why oxygen.
So we did note that the results at higher oxygenation changed, but we did see that there is a flash, or there's still like less oxygen depleted at flash rates than conventional. The question is how much. That's something to investigate in probably specific
the experimental
animal
data.
models.
I mean, maybe Charles can mention this, but the experimental data was, was pretty significant, the reduction in the effect.
Another question by
Yes, of
MCAT.
course.
That was again Joao.
No, well, Charlie can go first.
Okay, Charlie
It's fine,
first.
it's in the follow-up.
Well, I mean, to try to clarify that was the whole reason we proposed this oxygen depletion effect, but it's wrong. I mean, come on. We published a bunch of data since showing we get the flash effect at 2 gray, right? So the bottom line with the oxygen depletion hypothesis is, regardless how you tinker with the radiation chemistry measurements, most of the stuff done in the past was right. More or less, you need 100 gray to deplete 3% of oxygen, okay? We get the flash effect at doses well below 100 gray. So I'm not arguing the importance of oxygen. Someone told me it was important in the radiation response when I was a student. It's true, but it's not important for the flash effect.
Thank you.
Well, I'm going to go on a little bit in the same direction than Charlie, Mirna. What about if the oxygen is irrelevant after all, given what just Joao said, given your measurements, and given what is known in vivo in the tissue? And then I would argue that in your system, when you're using this cell buffer, your glutathione level is super high, 5 millimolar, nothing to be compared with anything relevant physiologically. And the same when you are adding the KCN compound to your system, to your cells, well, basically it's a toxic compound, so if your cells are dead, nothing does happen.
So this is based on our mechanistic studies and radiation chemistry. So this is what we are observing, so hence this is what we're trying to correlate. We're trying to understand at the chemistry level using our oxygen, and these are our empirical data so far showing, or we're trying to explain what is observed and correlating it with the flash effect.
So what about if chemistry would not inform us about the flash effect?
Of course, the flash effect is more complex, but we have to do our studies to understand it and,
Right. There's another question from online. Actually, Ron Levitt is raising 3 points regarding the flash effect model. First is, what about flash RT sparing of hypoxic tissues, or like skin, or compartments like stem cell niche? Maybe we'll go all. Second is, not all tumors are hypoxic. And the third, there's data presented last year in preprint and soon to be published showing greater flash RT efficiency against hypoxic tumors. How does that fit with the model?
Good question. So we do see still the flash effect at lower oxygenation, but the difference is not significant, and that's why you see tumor control when you're comparing it to the normal tissue, and the difference is much significant. I don't know if I answered the question.
Okay. Any other questions? If there are no other further questions, you can also find Mirna after the talk. And thank you very much again.
Thank you.